Hi, my name is Zahra Pakzad. I'm a PhD student at the University of British Columbia, and with my co-authors, I'm pleased to present on the Maternal Microbiome Legacy Project. As a brief introduction, studies suggest that infants born via C-section may be more susceptible to immune-mediated and metabolic conditions such as asthma and allergies. It's theorized that the maternal microbiome, particularly the vaginal microbiome, may play a role in this by helping establish the infant gut microbiome in relation to mode of delivery. However, this is not yet clearly understood. When conducting studies to answer this complex question, it's important to have large sample sizes and proper inclusion of confounders. I will be describing the methodological approach we took with the study and preliminary cohort characteristics. Our study population was a prospective cohort of women and people delivering at term at 3 large maternity hospitals in British Columbia, Canada, chosen to include a diverse population representative of our province. Vaginal swabs, infant stool, areola swabs, and breast milk samples were collected during 4 visit windows up to 3.5 months postpartum for microbiome analysis. Data collected via clinical chart review and participant questionnaires included extensive data about the pregnancy, labor and delivery, and postpartum periods. We selectively enrolled a total of 628 participants in 3 mode of delivery groups: vaginal deliveries, elective C-sections, and emergency C-sections. While we did have to close down recruitment due to the pandemic, ours is still one of the largest sample sizes of this type of study. As you can see on the right, we had a high rate of visit retention, with 82% completing all visits. In total, we've collected over 4,700 individual biological samples. Our team uses a CPN60 barcode sequencing for microbiome profiling, which has the advantage of higher species resolution compared to 16S rRNA profiling. Stay tuned for results from these analyses. I'd like to highlight some interesting learning lessons we've had while conducting the study. We found that in-person active recruitment was by far the most successful method of recruitment. Engagement with nursing and clinical staff across multiple hospital units was key to successful sample collection in a busy clinical environment. And our postpartum visits were done as home visits to be more convenient and accessible for our participants, which helped with the high rate of retention. Highlighting some of the demographic and clinical characteristics, mean age at delivery was 34, which is representative of our hospital's delivery populations. The percentages of self-identified ethnicities are generally representative of the population of BC, except for a smaller proportion identifying as Indigenous. For 40%, this was their first pregnancy. We had a small proportion who had delivered at home and a small proportion who had twins. Only 6 participants did vaginal seeding following their C-section, which is not unexpected given that Canadian guidelines and protocols at our centers recommend against vaginal seeding. 21% of participants had a positive GPS result, which is in line with our 3 centers' obstetric populations, and about 16% had gestational diabetes, with half treated with insulin. Looking at antibiotic usage during labor and delivery, 71% had antibiotics during labor and delivery, with 16% having 2 or more types. By far the most common indication was pre-op for C-section, where 94% had cefazolin pre-op, which is the protocol at our centers. Next was GBS positivity, with penicillin being the protocol, and those with chorioamnionitis were treated with a combination of metronidazole and cefazolin, which is the first-line therapy at our centers. This differs from other centers, particularly in the US, where predominantly clindamycin and gentamicin are used. On the right, as expected, all participants who had a C-section received antibiotics during labor and delivery, primarily cefazolin, and of those who had vaginal deliveries, 28% had antibiotics, with 71% having had penicillin. In summary, I've highlighted the Maternal Microbiome Legacy Project's extensive samples and data collected from a diverse group of participants to examine the extent to which the maternal microbiome contributes to the development of the infant gut microbiome. Stay tuned for further results. We'd like to acknowledge our research team, nursing staff at our 3 sites, our participants, as well as funding by CIHR. Thanks very much for listening.